Welcome back. Now, as a science broadcaster, I've been covering this subject that we're going to talk about next for a very long time, and it's always seemed to be a long time in the future. Finally, we seem to be getting to something that is very near indeed. I'm talking about stem cell derived islets, and with me to talk about the session that took place on this subject is Malin from the University of Lund in Sweden, Timo Otonkoski from the University of Helsinki, and finally Miriam Cnop from the Université Libre de Bruxelles. Miriam, let's start off with you. Where are we now?
Well, in my lab, we're using stem cells and the differentiation into islet cells as a very exciting model to study diabetes disease mechanisms and to test treatments. We're not working on transplantation, but it's an extraordinary model to work with because it is as close to the patient as you can possibly get.
Yeah, people have been talking about the transplantation bit, but not talking about actually this so important possibility of a disease model, and also a test bench, I guess, a test lab for new procedures. Timo, for you, where is this?
So, well, my lab has been working on the topic for quite a long time, and then after many years of error and trial and error, so finally I think we really managed to produce what I believe is maybe the best so far reported system to really get fully functional islet cells from stem cells, and we published that last year in Nature Biotechnology, and now we have been also training other people in Europe, particularly in using this. For example, Miriam's lab, and I'm really happy that it's going really fast forward now. And of course, we also know now that basically the similar technology and similar stem cell derived islets are already have entered clinical trials, although not yet in Europe, but in the USA. Already clinical trials ongoing. So I think we will be witnessing really fast progress and development during the next few years.
And how about you, Malin?
Yeah, so I'm really the novice sort of in this crowd. I haven't been working with stem cells for that long, actually. So my main interest is to understand how genetic variation actually affects the beta cells in type 2 diabetes. And for this, we're utilizing stem cells to try to understand this and genetic engineering, basically. So maybe we can improve the beta cell function through that.
And so why is this field suddenly come on so much when it was, I mean, you say, Timo, it's taken years of trial and error. What has changed?
Well, obviously the idea has been good all the time, but it was just for a very long time. It was an incredible challenge. So gradually, piece by piece, groups around the world learned how to really go from the stem cell to the next stage and the next stage. But it was still, until recently, not really possible to think that this would be a one-to-one model for the real primary islets and beta cell. But now I think we are very close. So what made it is hard to say. There's, like always in science, it's not just one single thing. It's a gradual progress and work by many groups and many people around the world.
And where do you see the main applications at the moment? Miriam?
Well, I think in general, I think stem cells, and I think Shinya Yamanaka got the Nobel Prize for a really fantastic discovery that has changed not only diabetes research, but so many different areas in biomedical research. I think the applications are innumerous. We're using it for some applications, as I mentioned, to understand disease mechanisms, understand why somebody develops diabetes. You can in those patient cells, test whether a drug works, yes or no. Is it going to be beneficial for the patient? And it's beautiful that you can test that in a dish before actually taking the medication into the patient. But it's also, for example, going to be useful to develop methods to do beta cell imaging, because that's also a human beta cell model that lends itself very well to that application. And obviously the cell replacement is going to become important.
So, Timo, let's look at the cell replacement. Do you think that, because of course lots of people think about this work purely in that vein, do you think that's going to be possible, or do you think the main application is going to be in drug discovery, in modelling, and in doing, testing before use of a drug in a particular patient?
Well, I personally have, for a long time, I have always said that I think the most valuable outcome will be the use in disease modeling and development of new drugs. I still do believe that the greatest benefit to the diabetic population will actually come from that work with new drugs. New mechanisms that can be clarified. But now, recently, when this really has started to progress also in the cell replacement, so I am also more optimistic. I do believe that it will go forward, but I wouldn't dare to say that it will be the everyday treatment for all type 1 diabetics. It would be too much, but who knows? I mean, I am not able to tell, but it will definitely also go forward, and I'm sure that, I'm hopeful that it will become an option.
I mean, certainly not something for all, but maybe for those patients who are completely refractory to all other treatments.
Yeah, well, I'm sure that if it is effective and safe, basically all type 1 diabetics would prefer it. I mean, if you are offered the possibility of getting one injection or whatever of cells into your body that then function for years and years without anything else, so I'm sure it is attractive. But let's see what happens.
Malin, for you, it's a journey of discovery.
Most definitely. I mean, for long I've been working on different animal models and cell lines, and I think finally we found models that, they actually come from the patient, and you can study like genetic mechanisms directly by microscopy, or to use as drug discovery, for instance, also, but actually to understand how genetic variation works, and that's like really, really small molecular changes. And I think that's super important, especially for type 2 diabetes, which is a polygenetic disorder. So that's sort of where we are going with this.
And we were talking about it earlier here about how type 1, we're now seeing it stratified into many different variants, and type 2 as well. So that work is going to be really important in guiding decision making on treatments.
Yeah, and I think also maybe for precision medicine, I mean, for instance, if you look at one specific variant and try to understand how that works, then maybe you can tailor make medication in that dish for those particular patients. So, I mean, that's also one application, of course.
So what's next, do you think? Take us forward 10 years, perhaps. Miriam, rub your crystal ball, read your tea leaves.
My crystal ball is out of order, I'm afraid. I think that there is a lot of methodological developments that has already taken place, and it's Timo's work that has really guided the progress in the field. It's not a model that I see as being readily adopted by very many laboratories in the world, because it is challenging.
And very complex.
It is complex, and it's not a technology that you implement like you might implement use of a cell line, or so it is labor intense and expensive research to do. But I think that the fact that you could take cells from individuals with a specific genetic variant or a mutation and test in the cells in a dish whether a drug is going to be beneficial, I think that is going to result in cost savings, because at the present time, what people do is to set up very big clinical trials based on a hypothesis that they have, but not having data that suggests that this hypothesis may be right. And I think that's really something that's in the coming years is going to develop further.
I mean, Timo, you've made a lot of the running here in developing the methodology. What still remains to be done?
Well, I suppose that the audience is probably most interested in this question of whether or not it can be developed into a therapy, the stem cell derived islets as a therapy for type 1 diabetes. And for that, if I try to look at the 10 years onwards from now, so the big question that has to be resolved obviously is how to be able to and we know that we can make functional cells, we know that we can put them in a patient, they will work, but how do we overcome the problem of their destruction by the immune system?
Yeah.
So because type 1 diabetes is an autoimmune disease, and we don't, if we want to apply this in a large scale, we don't obviously want to give it with heavy immunosuppressive medication. So that has to be overcome. And luckily, the one good side with this system is that we can easily genetically modify the cells. So that is the key, in my opinion, that to find the right type of genetic modification that will enable the cells to be used without the immune destruction. And then how to do it safely is the $1 million question. So, because obviously we are all the time playing with fire when we make them blind, we make them the immune system blind to these cells, and we put them in the patient, then they are potentially more dangerous, the cells. So there are many questions that need to be resolved, but there are now good avenues, good ideas of how to approach those questions. So I'm pretty optimistic that it will succeed in 10 years. Yes.
Malin, what's next in your field?
Well, try to find more genetic polymorphisms that we are interested to study and sort of apply them to the models that we're using currently with the iPS cells. We have many candidates that we're looking into currently. We've also started looking a bit on how to sort of modulate T cell recognition in beta cells from type 1 diabetes patients. It's very, very early on, but that would maybe sort of help in the field of transplantation ultimately. So yeah.
Well, thank you so much for talking to us at EASD TV about this. It's really appreciated because this is such an interesting subject and there's so much to say about it. So thank you. And a terrific session for you to watch on EASD TV. Bye for now.